A well-designed experiment has a clearly defined primary outcome, minimal bias, and maximal power. How do you accomplish this? Simple. Collect infinitely many independent observations in an unbiased manner and measure everything that could possibly affect the outcome. In reality, we are limited by the resources we have, as well as ethical concerns and possibly other constraints. Collecting more observations is costly and takes more time. And suppose you can answer a research question using 10 laboratory mice, then it is hard to justify using 20, 30, or even more. This is why study design is so important. How do you set up an experiment such that you can answer the research question to a satisfactory extent with limited resources? And even more important, how do you make sure that the conclusions drawn from your experiment are actually valid? For the purpose of this video, I will assume you're setting up an experiment to do a hypothesis test. This concerns any research question that you can reduce to a yes or no. Are men taller than women? Are bees more attracted to certain colors of flowers? And does tetracycline work as an antibiotic against E. coli? Of course, not every research question can be meaningfully reduced to a yes or no question. So in later videos, I will address more complicated designs and how to set up studies for different purposes than hypothesis testing. The more you know about statistics, the more interesting research questions you can consider. Nevertheless, most of the concepts I explain in this video apply to anything from simple correlation analysis to ANOVA, linear regression, and even far more complicated techniques. For the purpose of this video, the only thing you should really know is what a t-test and what a chi-square test is. If you're not sure about either, you can watch the videos linked in the description. First, I will explain what bias and power actually mean. Unbiased means that while individual observations may differ from one another, the result across a large number of samples should on average be equal to the value in the population of interest. Bias can occur due to all kinds of influences, including confounding, which is a topic for another video. But here is a simple example. Suppose you're growing bacteria under two conditions on agar plates to see if they grow differently, but you put both sets of agar plates in different stoves. Now, any differences in heat transfer between those stoves will bias the results. If one stove is very old and the other brand new, then this bias will be large. But even if both stoves appear to be the same, it is best not to take any risk of bias. Avoiding bias is very simple. If you randomly assign all the plates to the stoves, then there might still be a difference in heat transfer, but now that difference will average out across the samples in each group. Let's go over one more example. Suppose you're going to conduct a survey, and you're a university student, so out of convenience, you're going to distribute the survey to other students in your class. If you conduct a survey like this, then the results are biased. Your sample will be overrepresented by young adults with a certain level of intelligence and certain preferences or circumstances that cause them all to end up choosing the same study. This isn't necessarily a problem. If you want to know what your fellow students think, then this is the way to do it. But you can't generalize these results to the whole population. If you want to know what the average person out there thinks, you need to think of a way to collect samples such that any person is equally likely to be selected. You want a representative selection of your population of interest. So it is important that you specify what that population of interest is. Who exactly do you want to generalize the conclusions to? Then identify any potential sources of bias, and finally, use randomization wherever possible to nullify the effect of bias on the results. Any form of non-random sampling is likely to yield a biased sample. The other term I want to briefly explain is power. Statistical power is your ability to demonstrate an effect of interest. To understand what that means, we must first consider the difference between proof and evidence. The figure on the left shows that for any right triangle, a squared plus b squared equals c squared. As long as the triangle has a 90-degree angle, then this is true for any positive a and b. There are no counterexamples. In other words, provided its premises hold, proof is irrefutable. The same cannot be said for evidence. If Bob hates cats, Bob is near the cat, no one else is near the cat, and the cat is no longer with us, then maybe Bob killed the cat. But for all we know, the cat ate poison, got run over by a car, got a heart attack, whatever. Bob is just in the wrong place at the wrong time. The point here is that we would blame Bob only if we are convinced by the evidence. But no matter how compelling the evidence is, there is always some other possible explanation. In law, this is called proof beyond a reasonable doubt, but it isn't really proof, just very compelling evidence. Maybe you saw Bob stab the cat with your own eyes. Sure, maybe Bob is a certified cat surgeon. We could go on like this. You could question the account of an eyewitness, for example. I'm not saying this is realistic. I'm also not saying we should only convict people based on irrefutable proof, but it is important that you understand the difference between proof and evidence, because empirical sciences like biology primarily rely on evidence. This is no different when collecting data for a study. If you randomly pick 5 men and 5 women, you might observe that men are on average significantly taller than women. But you might also observe that there is no difference, or even that the women in your sample happen to be taller than the men in your sample. How do you know that what you see is not just a coincidence? These are just 10 individuals from a much larger population. We could collect more and more samples, but the problem will always persist. The groups in your sample may differ on average, while the groups in your population of interest do not. The opposite can of course also occur. In your sample, there may not be a significant difference, but that doesn't guarantee that there is no difference in the population. In statistics, we call these phenomena false positives and false negatives. If there is no effect in the population, but a test performed on your sample is significant anyway, that is called a false positive. If there is a difference in the population, but a test performed on your sample was unable to pick it up as significant, then that is called a false negative. And that finally brings us to the meaning of power. Power is your ability to demonstrate an effect with a sample if there is an effect in the population. The larger your power, the better your chances of demonstrating a true effect. It is equal to 1 minus the chance of a false negative. In practice, we don't know what the real answers are. We only have a sample, so you cannot actually determine if your result is real or a false positive or false negative. But even though we cannot know whether our result is correct, we can limit the probability that these errors will occur. To limit the chance of a false positive, simply be more strict about what you consider significant. By setting the level of significance alpha before seeing any of the results, you define a threshold for the maximum tolerated chance of a false positive. If your p-value is smaller than this threshold, then the test is significant at the level you specified. In many fields, it is common to use an alpha of 0.05, which means that you accept a 5% or 1 in 20 chance of a false positive. Of course, there's nothing magical about this number, and you can choose whatever value you think is appropriate as long as you do so before seeing the results of your experiment. If you're very worried about false positives, set alpha lower, like 0.01 instead of 0.05. If a false positive is not much of a problem, you can retain power by working with a higher alpha, like 0.1 for example. So that's how you limit the chance of a false positive. The chance of a false negative cannot be directly controlled. But of course, the more samples you collect, the closer you should be to the population, and therefore the better your chances of concluding a significant difference if there is a real difference in the population. How many observations you need precisely is something I'll discuss later on. But here's a rough guideline of all the things affecting the required sample size. If the difference you try to demonstrate is very large in the population, then of course you don't need many observations to also see it in the sample. And the other way around, if the difference is very subtle, you'll need a lot of observations to demonstrate it with significance. Second, if the extent to which individual observations differ from each other is large, then you will need many observations to see a significant result. And the other way around, if the difference is very consistent, you don't need many observations to see it. Though you cannot control the natural variance of a process, this of course also means that if you work very precise, the measurement error will be lower, and therefore you will need fewer samples than someone working sloppy. Lastly, if you're very strict about what counts as significant, then of course you're more likely to fail to conclude a significant difference. Let me explain. It might be tempting to set alpha very low, like 0.00001, and thereby reduce the chance of a false positive. But doing so, by definition, means that we are less likely to say that a difference is significant. After all, we're being more strict. So keep in mind that while lowering alpha will indeed reduce the chance of false positives, it will simultaneously increase the chance of false negatives. Now we know how to avoid bias, and we have some idea of what power is. To design a study for hypothesis testing, you should roughly follow this 5-step plan. First, you should define the primary outcome. This is whatever measure you will use to draw a conclusion. If you want to know whether males are taller than females, the primary outcome is the height of each individual. If you want to know whether bees are more attracted to certain flowers than others, the primary outcome could be the number of bees counted per flower within a given amount of time. And if you want to know whether some antibiotic can inhibit bacterial growth, your primary outcome could be either the number of bacterial colonies, or perhaps if it is very potent, the absence and presence of bacterial colonies on agar plates. Once you've written down what the primary outcome is, the next step is to convert your research question into a testable hypothesis. To address a scientific problem, the research question must be converted into something that can be expressed in numbers, or a simple yes or no. When you ask whether men are taller than women, there is ambiguity. Do you mean to ask whether all men are taller than all women? Or that men have a larger maximum height than women? Or do you perhaps mean that, on average, men are taller than women? So here are then our hypotheses. The average height of men is larger than the average height of women. Bees are, on average, more attracted to certain colors of flowers than others. And adding a certain concentration of tetracycline results in a different number of E. coli colonies compared to a control medium. Note how the second and third research question are already inherently more complicated. For the bees, which colors will you consider? How will you ensure that on average only the color differs between flowers? And for tetracycline, what concentration will you use? You could add a fixed amount of tetracycline versus a control, but you could also try a range of different concentrations. The latter is more work and calls for a more complicated analysis called ANCOVA, but will also give you a more interesting answer to your question, like, is there a concentration-dependent effect? So take this into account when designing your study. If this is your first experiment, I recommend keeping it simple. Only make the design more complicated if you already have an idea of how you're going to analyze that. One final piece of ambiguity remains. Namely, these hypotheses could be true or false depending on what we consider to be sufficiently larger, more, or different. If the mean height of men in my sample is 1 millimeter larger than that of women, does that count? Or say women vary a lot in their height, then how large should the difference be? To solve this problem, we instead define a statement that can be directly falsified, the null hypothesis. This is an unambiguous statement of no difference or no effect. So for the first research question, we begin by taking on the null hypothesis that the mean height of men is less than or equal to that of women. For if this statement is false, then on average, men must be taller than women, whatever taller means. For the second one, the null is that the number of visiting bees is equal, for if it is not, then bees have preference for a certain color. And for the last one, the null is that the number of colonies on either plate is equal. Or again, if the antibiotic simply kills off everything, you could have multiple plates of both the treatment and the control, and you could use the null that the number of plates with colonies is equal for both treatments. Now we can collect data as evidence against these statements of no effect. How that works exactly depends on the test used. But for any test, if we have enough evidence, we will have a large enough test statistic, which corresponds to a small enough p-value, such that we reject the null hypothesis and conclude that the difference is significant. By converting the research question into a null hypothesis, it already becomes clear what method could be used for analysis. You can compare means with some variation of the t-test, and you can compare frequencies with the chi-square test. Of course, there are more elaborate ways to do this, but in principle, this would be a correct approach. On to the required sample size then. The simplest answer to how many observations should I collect is as many as you can within a reasonable amount of time. More independent observations is more evidence, but it is always good to try and substantiate your sample size. For example, if there is no difference in means in the population, then your conclusion is so much more valuable if you know your study has sufficient power to conclude a biologically relevant difference. Estimating the required sample size will also give you an idea of the feasibility of your study. Maybe you need hundreds of observations to have sufficient power, even though you only have time for about a dozen. In a more extreme case, studies working with laboratory animals, let alone human beings, will in most cases require approval from an ethical committee. To gain approval, you will need to thoroughly substantiate your choice for the sample size. And even if you don't have to deal with any committee, as a scientist, you have a responsibility to perform ethical research. So if you ever work with live subjects, either read up a lot more on power calculation and simulation studies, or better yet, consult a statistician. For simple tests like the t-test and chi-square test though, there exists a strong and clear relationship between the power, how large the difference is, the variance, the significance level, and the required sample size. So you can simply plug all of these numbers except the sample size into a calculator and get a good idea of the sample size you need. In R, you can use the Power Package, PWR, which has a vignette that demonstrates many of its functions with examples. There are also many online tools out there for sample size calculation. Note, however, that while the relationship between these variables is exact, the numbers you plug in are often more of a best guess. How large the difference between groups is usually not something you know. That's why we're designing a study to find out. Nor do we know the extent to which individual observations differ from one another. You can search literature on your subject to see if you can find what other scientists saw for these values, and this is a great starting point. But keep in mind that these too are just estimates from samples. So I recommend plugging in several different sets of numbers to get a pessimistic and optimistic estimate of the required sample size. Another trick you can use is to just enter the smallest biologically relevant difference as the given effect size in these calculations. Finally, note that if you're going to use some online tool or the Power Package in R, you will often have to enter Cohen's d as the effect size, which is actually a combination of how large the expected difference between groups is and how large the expected difference between individual observations within a group is. There are guidelines for the interpretation of this value, but this is rather arbitrary and not everyone agrees on these. So again, try different combinations of inputs and see their effect on the required sample size. Then judge for yourself whether that sample size is something you can do given the resources and the time that you have. When all else fails, you can always determine the absolute minimal required sample size by counting the number of estimated parameters and adding 1. Here are some examples. In a one-sample t-test, you estimate 1 mean, so you need 2 samples. In a 2-sample t-test, you estimate 2 means, so you need 3 samples. In ANOVA, you estimate m, so you need m plus 1 samples. And in simple linear regression, we have an intercept and a slope, so you need 3 samples, etc. Of course, going with the minimal required sample size is unlikely to give you enough power, and getting a precise required sample size, as we just saw, can be quite difficult. So to reiterate, for any experiment, I recommend deciding on what is an appropriate investment in terms of time and other resources, and then collecting as many observations as possible. Throughout this video, I mentioned variance several times, the extent to which individual observations differ from one another. If this is large, you need more observations and vice versa. Together with the bias, it represents the uncertainty. So what causes variance, and how can you keep it as low as possible? First of all, there's of course the natural variation in any biological process. This is caused by a myriad of influences that we cannot measure or control directly. Think of minor differences in DNA between individuals, background radiation, random mutations occurring, random defects in genetic repair, and differences in anything really that we did not or could not measure. This is not something we can control, and that's why there is always uncertainty in our conclusions. But there are also variance components that we can, at least to some extent, influence. Think of the measurement error. Working more precisely, or using more high-tech equipment with greater precision and higher resolution will reduce your measurement error. But just like natural variation, measurement error cannot be completely eliminated. Your equipment cannot measure with perfect precision. Your pipetting skills may be good, but the difference between 1 microliter and 0.999 microliter is unlikely something you can control or even register on a typical pipette. Lastly, there's the variance caused by sampling error. Your sample is a limited selection of the population. It will always vary to some extent from the actual population. This is where random sampling and more elaborate techniques like stratified random sampling come in. But even if you did everything correctly, you are still dealing with a random sample. Your results are subject to chance. If you throw the dice once, you might roll 6. If you buy one lottery ticket, you might win the lottery. And if you draw a single sample from the population, you might just happen to have a sample that is not quite representative of its population. The last thing I want to warn you about before you design your study is pseudo-replication. When I talk about the required sample size, I mean the number of independent observations, or true replicates. Measuring leaves of 5 plants is not the same as measuring 5 leaves of one plant. The latter is called pseudo-replication, because these measurements are not independent. Some studies purposefully incorporate pseudo-replication. After all, if you measure multiple leaves per plant, you can average the results per plant and you will have a lower measurement error. But pseudo-replication does not count towards your required sample size, nor does it increase the power of your study. So always make sure that your experimental design will yield you enough independent samples. So in summary then, before you do any experiments or collect any data, define a clear purpose and a primary outcome of your study. Find the right statistical method for analyzing the results, and then try to substantiate your sample size based on the method you intend to use. Identify any potential sources of bias and try to keep the measurement error as low as possible. Only once you've taken all of this into consideration is it time to start performing experiments and collecting data. This will save you a lot of time and frustration in the long run. It will also make the statistical part a lot easier and your conclusions a lot stronger. If you don't know how to analyze something, you can always consult a statistician. But I highly recommend you do so before you start collecting your data. To quote Ronald Fisher, to consult a statistician after an experiment is finished is often merely to ask him to conduct a post-mortem examination. He can perhaps say what the experiment died of. Countless times have people asked me to help them with analysis, where my answer is simply, well, you cannot make any meaningful conclusions from these experiments. So make sure that your study design is on point. If you don't know any statisticians, and your supervisor or teacher doesn't know either, there's a great resource for help over at CrossValidated, or stats.stackexchange.com, where you can ask questions about anything related to statistics, including study design. That concludes all for this first video on study design. Thank you for watching.